So, hello everyone, my name is Kevin Clancy. I'm from ThermoFisher Scientific's Synthetic Biology Unit. What I'm going to be doing today is talking about how we use open source software and concepts that come from the synthetic biology community in order to develop various different kinds of tools and technologies that were then used by ourselves within our own laboratories or by our customers. So what I'm going to do mainly today is talk about genome editing tools and their applications. This actually is a really nice example for the synthetic biology community. In the genome editing tools actually fulfill many of the criteria that you would want to have for a synthetic biology tool. They're modular, they're scalable, they are orthogonal to each other, okay, and they're very, very capable of being modeled in software. And what we have done over the last couple of years is we've developed a whole series of different tools in order to streamline the process of cell engineering from what used to be weeks and months with an uncertain outcome down to a matter of 4 days with actually a very highly predictable outcome. So what is gene editing? Gene editing is basically going in and actually directly modifying the genome of an organism. Okay, it can be a bacteria. Typically we concentrate on eukaryotic organisms. And what we are doing in the process of doing that is we are either going in and we're adding a new piece of DNA, we're going in and we're placing a piece of DNA, or we are going in and we are removing a piece of DNA. And these are all valid gene editing workflows. So what people are typically doing in cell lines is they're killing genes in order to increase flow through a different pathway. They are modifying genes in order to be able to change the way the enzyme works, again, to modify what's happening in a pathway. Or they're adding entirely new genes to allow them to track what's going on in the cell or to make something new that the cell normally doesn't actually do. Now, the process of doing that in the past has been really quite difficult. So we have things like, say, mutagenesis technologies, where you're going in and you're hitting the DNA with the mutagen. Okay, those were very, very hit or miss technologies. You have things like meganucleases and more recently zinc finger nucleases, where again the programmability and the precision with which you're actually hitting parts of the genome are really still quite difficult. Okay, you have viral-based technologies, which I'm not showing here, which again, there's an awful lot of iffiness in terms of sort of like where you're actually going to hit your genome. Now, recently we've had a bit of a change in that we have new technologies, TALs, transcription activator-like effectors, our CRISPR-Cas9 system that we're now starting to implement, which are much more precise in terms of where they will actually hit and the consequences of where they will hit. And good, it's coming up. So, what we're doing now is we're taking these TALs or we're taking these CRISPRs and we're using those directly to engineer our genome. And we're using these to do a number of different kinds of changes to genomes. We're either introducing new pieces of DNA, okay, either gene addition or gene corrections, and we're using what's called homology-directed repair. So we are planning to go in, change the genome in a very precise way, okay, and that's going to allow us to insert or replace a piece of DNA in a predictable fashion. Or what we're simply doing is going in and disrupting a gene. Okay, that's non-homologous end joining. And in this particular case, again, we have a variety of different technologies that we can use to change our genome in a precise way. Now, the applications that we're basically applying these to are a wide variety of different applications which are used throughout both research laboratories, pharma, and large agribusinesses. We want to be able to build animal disease models so we can test drugs more effectively. We want to be able to do tissue disease modeling. Okay, so again, we can look at the consequences of new drugs on a disease cell line might actually be. We want to engineer stem cells. Okay, maybe one day, 20, 30 years down the road, we may actually be able to go into people and be able to treat them directly using these technologies. We may wish to go into gene therapy. And finally, we may wish to go into creating transgenic plants in a more efficient way than we've done in the past. And again, all of these are sort of like key applications for the cell engineering workflows that we're implementing. Now, both of these tools are the best fits, but the thing of it is you have to actually understand what tool you need to use and how you actually need to use it. So the tool that I'm going to concentrate on most is the CRISPR-Cas9 tool. In this particular case, you've got two main components. You've got a protein, Cas9, and you have a small stretch of gRNA, guide RNA, which is used to combine with that Cas9 protein. The combination of the Cas9 protein and the guide RNA allow you to very precisely say where in the genome that complex is going to bind. When that complex binds in your genome, then depending upon the nature of the Cas9 protein you're using, you're either going to get a double-stranded cut, or you're going to get a single-stranded nick, or in some cases we actually have a catalytically inactive Cas9, okay, which will just simply bind to the genome at that point and stay there. And that can be used for things like activation and repression. So these genome editing tools actually have a number of interesting properties. So as we work with these, we can see that we have workflows starting to develop. For in this particular case, we're using a DNA vector. The DNA vector has to be introduced into the cell. Once it's in the cell, RNA is made, protein is made, the complex is formed, and then it goes back into the nucleus in order to be able to edit the genome of that particular cell. In other cases, what we're doing now is actually introducing two RNAs, one for the Cas9 protein, one for the guide RNA. And again, what's actually happening here is both the RNAs are going into the cell, they have to be introduced, they're forming a complex after the Cas9 protein has been produced, and again, you're going in and editing the cell. Finally, we have a third variation, which is to turn around and simply complex the protein and the guide RNA outside of the cell and then transform that directly into the cell. What you're doing in this particular case is you're taking away the burden of actually making the protein and creating the complex, and you're going straight towards being able to edit your cell more directly. And so what ends up happening is that, as I said earlier, as you're using these technologies, you need to actually assess how you're going to use the technology and what the needs of your experiment actually are, okay? And what we're basically doing in our technologies is we're coming up with the experimental methods of how do you actually streamline these formats, how do you actually introduce them into the cell and what they can actually be used for. But while we're developing this, we're also starting to develop a data model of how to do these experiments. Many parts of this data model are actually reusable. It's almost a software design pattern. Okay, so again, what's very interesting about cell engineering is it is now approaching a point where we have the ability to take what is traditionally the realm of wet bench science and create software models for how wet bench science is supposed to behave and be able to use those as part of our design process. So, as we look at a synthetic biology view of how things are done, we can see the synthetic biology view concentrates on we've got individual pieces of DNA, our RNA, our protein, they're so-called parts. Parts have a functional outcome. Sometimes they're involved with replication of the plasmid. Sometimes they're involved in helping the plasmid survive and be selected within the media that the cell is growing in. Sometimes it's eukaryotic, sometimes it's prokaryotic. Other times, the cassette that you're using is actually being used to express the protein. Okay, but all of these are like in software design patterns. Okay, these are design patterns that can be used for how you actually do your experiment. Okay, and again, one of the advantages we're seeing of open source software, particularly for helping us to develop these kinds of tools, is sort of like providing models of how might you create a design pattern in software and allowing us to translate that through standards like synthetic biology open language into how would you actually model biology. So in this particular case, this is an example which was done by Chris Myers at the University of Utah. And what it is basically a CRISPR-based repression design. Okay, so we have basically a couple of different cassettes which are actually producing the CRISPR and the guide RNA. These are coming together to form the complex. They're then going towards their target, and then the target is basically having various functional consequences on reporter genes which are present within the cell itself. Okay, so again, what is interesting here is these models, okay, help us to be able to reuse our synthetic biology designs. So things that we would normally make in the lab, that we would normally do as a standalone GenBank file, that we would write up on our notepads, okay, and keep within a paper workbook, are now starting to look more like actually data models we would actually want to be able to reuse and compute over in software programs. So what I'm going to do is basically explain parts of our genome editing workflow, particularly where the software comes into this, and talk about how we use software for design, delivery, and detection. of what happens within you're using CRISPR designs. Now, in the first part, the design part, is basically all about making the guide RNA. Okay, so it's basically characterization and development of your parts. And for this gives you an example of our high throughput cloud design platform. So what we're doing is we're starting with coding sequences. We're trying to find regions which would correspond to a good guide RNA. We're evaluating those against the whole of the human genome, okay, in order to come up with A, the best designs, that B, are most unique to the target we're actually trying to hit. And we're then storing those in a database. Over here, you can see part of the compute infrastructure we had to put together to do this. So if you think about it, you've got 24,000 genes within the human genome, okay? Each one of those is between 1 and 10 exons at least. All of those have got pseudogenes to a certain extent. All of those may have repeats, okay? And what you have to do is basically be able to do, first of all, the identification of what is a good guide RNA target site. Then you have to have a very fast way of being able to screen that against the entire genome to find out, are there any other parts of the genome where you may have an off-target effect? Okay. Then you have to turn around and filter all of that data, and then finally store it within a database. So what we did is basically set up a way of being able to replicate many master node workers, okay, and be able to design a pipeline that first of all could be used to prototype designing all of these and doing the screen against the genome, and then subsequently be scaled up to be able to tackle any large eukaryotic genome. Okay, and where we're using software for this is basically throughout the entire process. We're using open source software as part of our data model for our database. Okay, so we relied very heavily on things like the UCSC data model, okay, and RefSeq data models. We're using softwares in order to be able to screen, first of all, against our genome. Okay, we're using Burroughs-Wheeler transforms, again, looking at how Bowtie was written, understanding the algorithm, then applying that to our workflows. And for the actual original description of the guide RNAs, again, what we're doing is we're reading papers which are in scientific journals. We're understanding the criteria for how to select those. And then we're basically going in and using those criteria specifically against our design needs for our technologies and tools. So without getting too much into the details of it, but if you want to know how to design a guide RNA, basically you've got a 3 base pair region called a PAM. What happens is basically you're then looking for a 20 nucleotide spacer region, which is upstream of the PAM. So first of all, look for any 3 nucleotide region which meets these criteria, and you can see a variety of these scattered throughout this particular piece of double-stranded DNA. You're then looking, the cut site is 3 bases downstream, or upstream rather, of the PAM site. So that's the cut site. And then what you're doing is you're evaluating for any one of these particular designs. First of all, this is the design we just came up with. Is it unique? Okay. Or does it in fact have another binding site which is exactly like it elsewhere in the genome? If it does, discard it. Okay. Does it have a unique binding site? Is there nothing like this throughout the rest of the entire genome? If it's unique to the entire genome, it's a great binding site. You want that. Then what we're doing is we're basically looking at thermodynamic calculations. And what we're doing is we're biasing towards possible other regions within the genome where you may have polymorphisms close to where the binding of the guide RNA and the genomic DNA take place. These are most likely to disrupt binding in the first place, and hence you won't actually get any Cas9 CRISPR complex binding to that piece of genomic DNA. Instead, what we're doing is we're looking for sequences which have either got, few to no polymorphisms down here, and we're looking for sequences which have got sort of like, ideally, any biases in sequence binding here. Okay, and this is really what we're using to do the guide RNA selections. Then what we're doing is we're scanning our model, okay, and we're looking for binding. We're looking for mismatch locations, GC content, and so on and so forth. And we're basically asking the question, how likely is it a given CRISPR and a proposed binding site could produce an off-target cleavage? And as we go through our pipeline, we then built up a database of possible designs we could actually use. So what you can see here is in the human and in the mouse, we have approximately 400 to 500,000 possible designs that pass all of our screening criteria. Of those designs, what we see is basically CRISPRs and TALs approximately have 18,000 different designs which are in common. We can hit 18,000 genes. And in fact, this very nicely overlaps with an older technology, siRNA, which has been used for many years by the cell engineering community to temporarily knock down the expression of a gene. Okay. And what we can also see is sort of like there is an awful lot of overlap of regions where everything is shared, all three technologies work, and we can see that there are some places where we have overlaps purely between CRISPRs and TALs. We're not able to find any siRNA designs. So in actual fact, these technologies are also giving us benefits into genes that we couldn't hit before through older technologies like siRNA can now actually be accessed. Now we go through to the second step, which is basically starting to deliver this in and actually use it in the cell line. So what we're doing here is we're now starting to come up with what we call validated workflows. So the idea behind a validated workflow is you've standardized not only the design of the guide RNA and how to put the guide RNA together with the CRISPR, but you've also validated every aspect of how to actually do the experiment, all the way from growing the cells, all the way through to preparing the Cas9, all the way through adding the two together and incubating it, and then turning around and being able to analyze whether the experiment has actually worked or not. Okay, so these are all part of what we now call our validated workflow. And again, we're using software throughout this. So where software is coming in important is now actually in the process of sort of like being able to manage all of our designs, which cell was hit by a particular experiment. It's in the process of turning around and actually seeing, did we actually get a guide RNA? Okay, is it amplifying correctly? And more importantly, once we actually go to do the experiment, does the experiment actually work?